Hi, everyone. Welcome to Jack's Career Chats, a series where we highlight the diversity of jobs, skills, and opportunities in the world of biomedical research. Today, I'm talking with my guest, Suleiman Bozal, who is currently working as a research assistant in the single-cell bio division at Jack's, the Jackson Laboratories campus in Farmington, Connecticut. Suli, thank you so much for taking the time to join me today.
Yeah, I'm happy to be here.
So tell us a little bit about what you do as a research assistant in this single-cell bio division at Jack's.
Yeah, so I work in a single cell biology lab, and um, I'm a research assistant there. So what I do is, conduct experiments and aid other scientists and researchers in various different projects. So I'm involved in a couple different ones. Right now, we're working on developing a high content method to screen organoid models, which are cells that are grown in 3D, and a lot of these are derived from cancer samples. And basically, what we do is we're trying to develop a system that will allow us to screen a bunch of different drugs to see what is effective at treating these cancers by targeting these organoids. I work on another project as well, and that's an endometriosis related project. So there's a scientist in our lab, Elise Courtois, who is interested in endometriosis, and we're using a single cell approach to understand the disease a little bit better and to develop some or to find some biomarkers that may be able to help us better identify the disease and hopefully treat it in the future.
Wow, those are two really interesting but very different projects.
Oh yeah, for sure. Different stuff, different techniques that we use, but it's really, really cool, very fun. I like working there.
Can you talk a little bit about the organoids? So that might be a new word to some of our users, to some of our viewers. So those are like you were mentioning 3D. So like instead of having cells on a dish, you're making them kind of aggregate, or how does that work?
Yeah, so essentially, yeah, you're right. A lot of cells are grown in 2D. So basically, you have a tissue dish that the cells adhere to, and then they spread out as they grow, and you add media so that they can grow, and you feed them basically. What they discovered some time ago, I don't know exactly how long ago, is that if you provide a mechanical barrier to these cells, we call it, it's a 3D matrix, and basically these cells, once they sense that mechanical barrier, different genes are activated, and that allows them to gain a polarity and grow in a way that's not 2D. So they grow as sort of like balls almost, and that's what the organoids are.
Cool. So you're like giving it a structure, like a framework to kind of.
Yeah,
Okay.
exactly. So this 3D matrix that we use is we embed the organoids in them. So we have like little, really small clusters of cells, but they can also be grown from single cells. And basically, once they start dividing, these cells can aggregate in a way that makes them, look like a sphere. And they have some cool, interesting properties when compared to the 2D monolayer of cells that a lot of people are used to.
Yeah. So then what's the advantage of having them, aside from being like a cool 3D structure? Why would you want to do this instead of just having them on the Petri dish in the single layer?
Yeah, so growing them as organoids, as a 3D structure, it's more physiologically relevant. Cells, our bodies aren't growing in 2D. We're not on a plate. So from that perspective, it allows us to model the human physiology a little bit better.
Yeah, that makes sense. So if you're trying to look at like a disease or these tumors or something like that, then mimicking the structure that is actually in the body is the best way to study it.
Exactly.
Yeah. And the other project you said was an endometriosis focused project.
Yep.
And
Um,
what are some of the things that you're looking at for that?
yeah, so endometriosis is a gynecological disease that affects, I think, around 1 in 10 women. It causes infertility and pain in these women, and it's really poorly understood, and there aren't too many treatment regimens for it. So what we're trying to do is understand it on a single cell level. So the lab is called the single cell biology lab, and one of the techniques we employ is, single cell biology, which is analyzing um, uh, these cells at a single cell level. So a lot of the experiments that are traditionally run, they have you know uh, you're kind of aggregating a bunch of cells to look at either the DNA or the RNA in them, and it's all then pooled together. And so when you analyze them at a single cell level, you're isolating each single cell and looking at the differences between cells that may exist in a sample. So you're able to analyze a lot of the heterogeneity between the different cell types in the tissue that you're looking at. So for this endometriosis project, what we're doing is looking at the lesions that grow in the endometrium and comparing them to normal tissue that comes from either a patient that doesn't have endometriosis, or we're comparing it to an endometriosis patient that where the tissue looks normal, so it's not a lesion. So looking at the differences and trying to identify what makes the lesion a lesion, and hopefully identify things that will help us be able to identify the disease early on, and again open up some treatment regimens earlier on, or some cool stuff, novel treatments, which maybe in the future.
Yeah. And what are some of the things that you do day to day in the lab, like in terms of techniques, and what do your days look like, or your weeks, if you don't
Yeah,
have
so
a
I
day?
think my days and weeks vary very, it's a very wide range of things that I do. A typical day might consist of going into the lab and taking care of my organoids. So basically feeding them, making sure that they're healthy, passaging them, which means when they grow and there's too many organoids in a dish, or there's too many cells in a dish, basically what you have to do is passage them, which means divide them, maybe take a fourth of the organoids that are in a plate and then put them into another plate.
Because they're constantly
So
growing, so you got to like make
exactly,
space for them.
exactly. And preserving those samples so we can grow them later for another experiment. So a lot of that. In addition to that, it's the system that we're developing that will allow us to screen these drugs across the organoids. There's a lot of optimization that goes into that in terms of understanding how we can have our workflow optimized. So basically just conducting experiments that will help us understand the best way to go through the entire process of growing the organoids, plating them, drugging them, and then our final readouts, which basically will tell us whether these organoids are living or dead, or what sort of state that they're in.
So it sounds like a lot of planning in addition to all of the work in the lab.
Yeah, it is a lot of planning. I mean, in addition to the planning, at the end of each experiment, there's a lot of data to analyze. So that's the other part. Sometimes I'm not in the lab. I'm actually at my computer trying to understand the data and trying to understand what might have worked best and what we can do differently next time.
Oh, okay, so you're working in the lab, but you also have some computer-based work. What can you describe what types of things you do on the computer? Like, what does that analysis look like?
Yeah, sure. So basically, let's say that we have a plate. It has 384 wells. In each well, there's a certain number of organoids, and we've treated this 384 well plate with different drugs. So in each well is a different drug combination. So at the end of the experiment, after 6 days of treatment, as in we've treated it, we waited 6 days to see what the response might be like. To understand if the cells are dying or not, what we can do is cell titer glow assay is what it's called. But basically, it's just measuring the ATP in each well, and that will give us an idea of whether or not the organoids in that well are dead or not. And so basically, once you have this readout, which we get by putting it into an instrument that will shine light at each well, and then it'll luminesce, and that luminescence is related to how much ATP is in there. So understanding what each well, how much it luminesces, will give us an idea of whether the organoids in it are dead or alive. So basically trying to map each well back to the original treatment regimen, understand the differences between the wells, and eventually between the different treatments, and understand what worked or not. So a lot of analysis goes into understanding that, and again, figuring out ways that we can improve upon it.
Yeah, cool. Thanks for explaining that process. And it's really interesting to hear like all of the physical lab work that goes into it, but then like all of the data analysis processing on the tail end of the experiments.
Yeah,
Kind of think
absolutely.
about like, oh, working in the lab, like working with your hands, but then you got to make sense of the data. So it's great to hear like how you're doing that.
Yeah.
So the lab that you work in, this single cell bio lab, is not like maybe a traditional lab that people think of run by a professor with like kind of one research goal. this is like a service type lab that works with a lot of different other laboratories for their individual projects. So it
Yeah.
sounds like that's probably a reason why you get to do these different types of projects.
Yeah, absolutely. I mean, the lab itself, there's it's sort of split. There's a service aspect to it where I guess we, work with other scientists to help them with their experiments, and then there's a research component as well. So the PI Paul Robson, or Dr. Paul Robson, that runs the lab, he also does his own research. So he has his own research interests that he works with other graduate students to um, to pursue. But yeah, the as far as the service side, there's a lot of cool instruments that I think we have access to, or easy access to, because it's a service lab. So yeah, definitely a lot of cool things that we get to do.
So how did you come to this position? Is this the job that you knew you wanted to have, since you were younger, or what got you to this position that you're in now?
Yeah, so I made the decision. The decision itself was made at the end of my undergraduate career. I decided to take some years to pursue research after graduation, before applying to graduate programs. So that's what I'm doing right now. And before that, sort of, I'll start in, I guess, high school. I didn't really know exactly what I wanted to do. I knew that I liked science. I had a curiosity towards it, but it was really during my undergraduate years. I took a class that sort of gave an intro to research, and I was really fascinated by, I guess, the way that the researchers and the people that presented to us were able to use a lot of the creativity and a lot of techniques that we had sort of been exposed to create solutions to problems that affected people. That to me was fascinating. I had, when I was in high school, and I thought about research, I didn't really think too much about, like, exactly what went into it. It was sort of like a black box to me. I felt like I understood some of the concepts that I was learning, but I didn't really see how it could be applied to discover something that was completely unknown, or to develop something that was totally new. And when I got to college and I was sort of exposed to that a little bit more, that's what really got me interested in, I guess, the research.
Yeah, I love that you said that. And I think that's so true. Like, one difference between high school research and kind of research beyond is that oftentimes in high school and class, like, there's a known thing that you're trying to get at, right? I mean, it's
Yeah.
kind of, the constraints of the experiment, because you're going to learn something, what the end goal is. But in real research, you don't know. You're trying to discover something new. And that's super exciting.
Yeah, absolutely. I mean, I think that in high school, I think it's easier to be, I guess, bored by what you're doing in a lab setting, because you know what you should get, or that you're expected to get a certain answer. I guess when you take that away and you're doing research in, I guess, unknown topics, it's kind of exciting. And so I guess that's actually one thing that I would say to myself in high school was, what you're learning is basically the foundation that will allow you to make these sort of findings later on, and you're just building I guess, the backbone of your knowledge, so that you can do this sort of research later on.
Yeah, absolutely. Another thing that I liked that you said was that you saw research as an opportunity to help people. And I think a lot of people get into science and medicine because, we want not only to learn something, but to help, to do better, and to help people, and to make progress. And that research affords you this opportunity to do that.
Yeah, absolutely. That's, yeah, exactly what um, led me to pursuing what I currently am.
So now that when you were an undergrad, you kind of saw all the potential of research and you got really into it. Did you have any experiences that really helped put you on the path of research, or give you more experience in research?
Yeah, yeah. So I was lucky in undergrad to, the class that I mentioned that was sort of exposing us to the class of these topics. I approached the professor because, I was interested in some of what she was doing, and I joined the lab that she was a part of at UConn. And so, after gaining some experience and going a little bit independently in terms of the research projects that I was involved in, I got involved in other various summer research programs. So, across the country, there's a lot of these summer research programs that will allow undergraduates to come in and learn from the scientists there and to contribute to a project. So, I got my feet wet in a lot of different areas, and I sort of pursued whatever I was interested in at the time. So I have some, different research experiences that aren't, all exactly related to what I'm doing now, but I guess it was a good way to learn what else is out there and to find, my interests and to develop more skills, I guess.
Yeah, definitely. It sounds like exactly that. It gave you, just a breadth of experiences to really be able to decide what you wanted and what you liked and what you didn't like.
Yeah, exactly.
And how did you find out about these experiences? Was there like a career center or research center? How do you know about these opportunities?
Yeah. There was, there's definitely a career center, and you can find a lot of these opportunities there. The first one I attended was actually at UNC, and that was sort of proposed by one of my professors, my advisor, and he mentioned the program and decided to apply to it. I was a structural biology and biophysics major, so the program was very oriented towards biophysics, and that's the first one that I had, and then. Later on, I actually did the summer student program at the Jackson Laboratory. There I had, saw through, I sort of knew about the program through just because of the partnership between UConn and JAX. And I decided to apply for it. I gave it a whirl and I got in. I was accepted. It was really, really exciting. And yeah, that was, I think, at the end of my junior year, I attended the summer student program.
So some of these summer programs, like you said, you have to apply and be admitted. So
Yeah.
they're likely competitive. Do you have any advice for any students that are looking to pursue some of these summer research opportunities?
Um, yeah, I think so. I think everyone's path is different in terms of what they might be interested in or what their background is. I think having a good understanding of what makes you you, and understanding how you can pursue those interests and getting an idea about the possibilities that you may have after the program, that can, I think, help you in writing a compelling application. And then I guess the other aspects are, performing well in your classes. That's very important to demonstrate that you have that sort of uh, background that will allow you to conduct the research effectively when you get to the program.
Yeah, thanks for sharing that. And for these summer programs, especially if they're far away, you were at UConn, but you did something at UNC, North Carolina. So was there funding involved? Like, how do you kind of pay for yourself to be
Yeah.
off for a summer and to do research or to stay somewhere else?
Yeah. I think there's a lot of students who are lucky enough to, just ask a professor and, mention that they don't really need funding because they can stay at home or whatever. That wasn't really always an option for me. So, before applying to these programs, I worked as, lifeguard during the summers, and I wanted income because I just did. I didn't really, I didn't want to pursue something where I wouldn't be funded. And so, yeah, a lot of these programs are funded. You're not getting paid an extravagant amount, but it's comparable to what you might be doing as a regular college student. And so that was really great for me. I was funded well during both summer programs at the UNC Chapel Hill and at Jax. So yeah, it was fun
Yeah,
there.
that's great to know. I mean, that can be a huge relief to be able to have some funding during the summer for these
Yeah.
opportunities. And so, were those experiences helpful to you, like to decide that you wanted to do research? It sounds like since you had those research experiences and now you chose a research position and not something else, were they like really formative for helping you stay on this research path?
Yeah, they really were. I think when I was in these summer programs and during my research at in undergrad at UConn, I sort of saw that the research that I was doing, I saw the potential that I had to, inform other researchers, and it had the potential to affect people, and that's what really drew me to research in general. It wasn't the funding. It's more like what I would be fulfilled, um, or what would fulfill me in my career. So that's what I guess led me to pursuing this. And I guess there's stages as well in terms of you're a student and then you gain these experiences and you're sort of gaining an idea of what will make you fulfilled, what you want to do. And that's truly what led me to pursuing this position after graduation.
Yeah, and so you mentioned also like taking some time to work before you go to graduate school. So you're interested to go to graduate school after?
Yeah, I'm applying right now. We'll see
Oh,
how it goes.
wow.
Yeah,
Good luck.
thanks.
It's
Thank
a
you. Yeah,
nerve-wracking experience.
yeah, I'm sure you know. Yeah,
Yeah,
absolutely. It's been sort of a stressful process just because of everything that's going on as well. A lot of interviews are virtual, which it'd be nice if I could see the campuses, but I guess we're adapting to what's going on.
sure. Yeah.
And so yeah, that's again like the next stage for me at least. I hope to go to graduate school.
And can I ask, why did you decide to go to graduate school? So you liked research. It sounds like you have a job that is, fitting these skills, and you're able to help people with the work that you do. Why did you feel the need that graduate school, taking on the next step in your education, was important for you?
Yeah, I think that having after you graduate from undergrad, having that extra degree or those extra degrees will they'll essentially open doors for you to get you where you want to go in terms of running your own research lab or getting into a position that's a little bit more I guess higher, and being able to make decisions and to pursue your own research interests completely independently of what other researchers may have lined up for you. I think that was that's what compelled me to go, I guess, the next step in general.
Yeah. And what are some things that you're considering about graduate school in terms of, like, how are you making your decisions? Not that you've made a decision yet, but just like,
Yeah.
which schools to apply to or what programs to apply to. Just curious.
Yeah, so I'm applying to MD PhD programs. So I guess in general, there's less of those programs than there are, in general graduate programs or medical schools. And so there's a few different aspects that go into deciding for me what makes an attractive MD PhD program. One of them is, I guess, the sort of um, the strengths of the school in terms of their graduate school and the research of the faculty um, that teach there or that do research there. So that's a start, I guess. And I would want to go to a school that has researchers whose topics or research that I'm interested in.
Right. You want to
So
work on something that you're interested in.
that I think is cool. Yeah, basically. I guess the other part is also related to I guess the medical school aspect of it in terms of who am I going to be able to learn from in terms of the patients that maybe that I may have the opportunity to interact with. Um, that's a factor. I guess funding is also a factor in this case. It would be nice if I was funded. And a lot of MD PhD programs they waive your medical school tuition, which is great, so you're not in as much debt, and a lot of them actually pay a stipend, so you get paid to go to school essentially. I guess the commitment is long, so it takes longer to graduate from these programs because you're getting a medical degree and a PhD, but I think the rewards from it are ultimately worth it.
And so what will you be able to do after that? You'll be like a physician scientist, so be able to practice medicine and do research?
Yes, exactly. That's the goal. So essentially, yeah, being able to practice medicine, to see patients, to learn from them, and then use a lot of what I learn from that medical education and from interacting with patients to inform future studies in terms of research. So that's the ultimate goal. So
Yeah. Yeah, no, that's really exciting. And I just have a question about your, like, skills and interests. Like, have you identified things like in your own kind of personality, things that you're good at, and that are helping to shape like these decisions in your career path, or
um,
like the type of work that you do?
I think so. I think one trait that I think I have is sort of like adaptability and a little bit of resilience. I think I've
Definitely need that in science and in
yeah, I think I've a lot of
experiments fail.
yeah, absolutely. I think I've put myself in a lot of situations where I knew I would be exposed to things that were new to me, and I knew where I knew that I'd be challenged in those situations. And I think having I guess the will to do that is important, because you want to ultimately challenge yourself and challenge what you know, and hopefully learn from either your mistakes or from your successes.
Yeah, so along those lines, do you have any words of advice for any aspiring physician scientists, MD, PhD students, anyone pursuing research or just kind of starting to think about this as a career path?
Um, yeah, so I guess in terms of advice, I think it would be to sort of do what I was mentioning before in terms of, I guess, sticking yourself in situations where you know you're going to learn something, even though it may be challenging for you, even though you may have to read that research article when you're starting out in the lab, 10 times over. Um, I guess that would be my advice. So, um, also another I think advice that I would have to high school students is to not let your lab classes dictate whether or not you're interested in science. Because I think what I've learned is that um, it's a lot more uh, it's a lot easier to become bored by what you're doing in, I guess, a lab class than it is to be bored by what you're doing in a real research setting where you're sort of trying to discover something new.
All right, well, Suli, thank you so much. This was a really great conversation. I loved hearing about your research and your really interesting career path, and wish you
Yeah.
the best of luck in grad school and beyond.
Yeah. Thanks, Christina. It's nice being
And
here.
thanks everyone for watching to this episode of the Career Chats. Don't forget to watch more. And until next time, bye-bye.